Okay, so we'll be discussing about the monoclonal antibody. So monoclonal antibody is an antibody that is very specific only to an antigen. This is monoclonal. So mono mean to say this is an antibody that specifically react with a specific antigen. Okay, so monoclonal antibody production has been produced here through our hybridoma technology. So when we speak about the hybridoma technology, you try to fuse primarily the spleen cell coming from your laboratory animal containing your B cell or B lymphocyte plus the myeloma cell or the cancer cell. This was then developed by Kohler and Milstein. Okay, so we have here the steps for the antibody production for the monoclonal antibody production. So first one, we have here a specific antigen is being injected to your laboratory animal. That can be either a mouse, a mice, or laboratory rat. Then as a product of antigenic stimulation, so the mouse immune system here, specifically the spleen of that, try to produce an antibody. Okay, or the B cell, okay, or antibody that would be very specific here for the antigen that has been injected with your laboratory animal. So all you need to do here after a week or 2 weeks here, you try to harvest the spleen. And the spleen here contains already our B lymphocytes that produces antibody already, specific for the antigen which has been injected to your laboratory animal. And with that, you just need to harvest the spleen. Okay, and then try to fuse that one with your myeloma cell or cancer cell, okay, by the action of your polyethylene glycol. So as an outcome, you can have the following product of that. You can have a fused na spleen cell, so spleen cell plus a spleen cell, myeloma cell plus your myeloma cell, or you could also have here your myeloma plus your spleen cell. But of course, we are only after of this myeloma plus your spleen cell. And then, allow this one to grow in your hot medium. Okay, the hot medium here is your hypoxanthine, aminopterin, thymidine medium. Okay, so allow to grow on that culture media. So in order for that to grow in a culture media, so it need to produce or synthesize its new nucleotides. Okay, so that's the requirement in order for that to grow in our hot medium. Okay, when you are synthesizing our nucleotide, so there are two pathway for that. We have here the de novo synthesis of your nucleotide. So pag sinabing de novo is a new production of your new nucleotide. When speak about naman salvage pathway, you are just recycling the byproducts of your nucleotide. Okay, coming from, for example here, coming from your dead cells. So when you have your dead cells, so the nucleus of that contains your purine and your pyrimidines, which are the byproducts. I mean, those are our products, which those are the materials which are needed primarily to produce here our new nucleotides or to produce or to recycle your nucleotide. Okay, but if you are utilizing that pathway, the salvage pathway, you'll be needing here the enzyme. Hypoxanthine, guanine, phosphoribosyltransferase, transferase, or you could also have here your thymidine kinase. In order for you to recycle those cells which has died already, and then try to release here the purines and pyrimidines in order for you to have here or produce a new nucleotide out of your mga nasirang mga cell natin. But when speak about your de novo, so this is the production of a new nucleotide. Okay, so if you have here your spleen plus your spleen cell na fusion, that one typically will not grow in our culture media. So X na ito, myeloma cell plus our myeloma cell on the other hand cannot also grow in our hot culture medium. It's because here, okay, this myeloma cells do not have the enzymes, okay, that in order for you to synthesize your nucleotide using this pathway. You could not utilize here the de novo synthesis because of the action of your aminopterin, which is part of the reagent component of your hot culture medium. So, wala kang choice dito. Hindi mo ito magagamit because again, that's already being blocked here by your aminopterin. So, ito na lang ang option mo in order for you to produce your nucleotides. But then again, if you have your myeloma cell, the myeloma cells here could not synthesize the enzyme. So, X din ang ating myeloma cells, of course, because they don't have the enzyme. Whereas our hybrid, the combination of our myeloma cell and our spleen cell can synthesize the enzyme. And therefore, they can produce new nucleotides, and therefore, they can able to grow in our culture media, in your hot culture media. So, after that one, you just need to harvest. So, ang maiiwan na lang ito, myeloma plus your spleen cell. Okay, that would allow you to harvest that one. So, since this hybrid here is a combination of your myeloma and your spleen cell, the myeloma cell here is the cancer cell, and one of the property of your cancer cell here able to grow or to divide at a faster rate, and at the same time can grow indefinitely. So, with that, you can able to produce so many products of your monoclonal antibody. Okay, you just need to harvest, you just need to concentrate, and what you have already would be your monoclonal antibody. So, for the application of the monoclonal antibody, so we can use this, okay, as our reagent for your test kit. Like for example, for the detection of your pregnancy test, for the detection of your beta-human chorionic gonadotropin. So, the test kit membrane can be impregnated by your monoclonal antibody directed towards your human chorionic gonadotropin na antigen in your sample. So therefore, that one is being utilized or applied on this. Another one, monoclonal antibodies could also be used here as a treatment for different autoimmune disorders. And of course, we have here your Crohn's disease and many others. Okay, next we have here the major histocompatibility complex. The former name of that is your MHC, but today it's being called here as the human leukocyte antigen. Okay, so basically because this one first isolated from the human leukocyte. Okay, so the major histocompatibility complex is one of the determinant of the success of our transplantation. Okay, so in order for the recipient and the donor here to be compatible with one another, then you need to have a compatible MHC or the human leukocyte antigen. That's why being called here as the major histocompatibility complex, because again, the transplanted tissue from your donor to your recipient should be histocompatible in order for you to have your successful na transplantation. Okay, so the gene here coding for the MHC is located in chromosome number 6 on the band 21 in the short arm of that. So this one is your chromosome number 6. We have the short arm and we have here the long arm. So there are three regions here. Actually, there are three regions on which your MHC molecule here is being encoded or could be found. We have here MHC class 1, MHC class 2, and at the middle portion of that, the chromosome number 6, you can find here your MHC class 3. Okay, so we have here the overview of your major histocompatibility complex. The MHC class 1, okay, is found here. So your MHC class 1, there are three regions for that, divided here into your HLA-A, HLA-B, HLA-C. Okay, this is used here primarily for the antigen presentation. Okay, so we'll be discussing that one later on kung saan niya ipipresent ang ating antigen. Okay, and this one, MSC class 1, usually found on the surface. Nasa labas siya, cell surface siya ng ating mga cell. MSC class 2, on the other hand, so this is located here in your HLA-D, divided here into your HLA-DR, DP, and we have also your DQ. The same with your MSC class 1 is also involved in our antigen presentation. And this is also found here on the cell surface, nasa cell membrane, cell surface. Whereas our MSC class 3, on the other hand, is being secreted in our plasma. So, I mean to say this is not found here on the cell surface. So, this one here, MSC class 3, try to code here for the production of your complement components, like your C2, your C4 of your classical pathway, and we have also here the factor B of our alternative pathway ng complement natin. And your MSC class 3 is also coding here for the production of your tumor necrosis factor. Okay, your tumor necrosis factor and our complement here do not involve in your antigen presentation. So they have another function. They have a separate function. Complement, so tumor necrosis factor, so they are not involved in our antigen presentation, unlike with your MSC class 1 and our MSC class 2. Okay, for the inheritance of our MSC here, so this is your primarily inherited as co-dominant allele. So mean to say one gene, one set of your MSC here, MSC class 1, 2, and 3 from your father, and another set here from your mother. So this co-dominant allele. Okay, so we have here the Okay, so we'll be discussing each of your MSC here, primarily involved in our antigen presentation. Let's include your MSC class 1 and MSC class 2. Okay, first we have your MSC class 1. So the MSC class 1 are found on your MSC class 1 found here on our nucleated cells in our body. So those cells having the nucleus, so of course we have your WBC, except your, the biomature RBC, don't have the nucleus. Okay, so it is where you can find here your MSC class 1. Okay, this is also not found on your hepatocyte, in your neural cell, muscle cell, sperm cell. Okay, what would be the implication of this one since they are not found on your hepatocyte in these mga cells? So basically, like, for example, the patient would have a liver transplantation. You don't need to check for the MSC class 1. Okay, because in the first place, wala naman siyang MSC class 1, diba? So, safe tayo with your MSC class 1. In terms of the compatibility between the donor and recipient is because na ang ating hepatocyte are not, hepatocyte here is not expressing your MSC class 1. Okay, so among our nucleated cells in our body here, the one found here to have the greatest concentration of your MSC class 1 would be our lymphocytes. Okay, so we have here the three loci or location. We have HLA-A, HLA-B, and HLA-C. So they form part our MSC class 1. Among the three here, HLA-A found here to be the greatest concentration. Again, what would be the implication for that? So, in order for you to have a successful transplantation between the donor and the recipient, the donor recipient should eventually be histocompatible or should be compatible with their HLA-A. Because again, that one is among your HLA here, your HLA-A is found on the highest concentration. So pag hindi sila compatible, of course, it will have a greater chance of rejection. Okay, as with the function of our MSC class 1, so before we go with the function, so we have here the structure of our MSC class 1, so it's made up of your alpha and your beta globin. The alpha globin here is made up of the alpha 1, alpha 2, and alpha 3. And we have here the beta globin, we have your beta 2. Primarily, that's the light chain component that forms part here of your beta 2 microglobulin. And we have also here ang ating peptide binding cleft or groove can be found here between your alpha 1 and your alpha 2. So this is a groove which try to capture the antigen, and that one we're going to present the antigen. Okay, for the function of our MSC class 1, so this one is try to involve here in the antigen presentation of what antigen. So we have here the virally infected cell or the tumor infected cell. For example, we have here your virally infected cell or a tumor-infected cell. So, ika-capture ito ng ito. Okay, then ipipresent niya dito sa labas. Okay, for example, dito na ang antigen natin, which is your viral pathogen or tumor. So, ipipresent niya dito sa labas. Okay, so this should be kasama ito, ipipresent dito ng MSC class 1 natin para makilala siya ni T cytotoxic natin. Okay, your T cytotoxic is your sedate positive. What associate with our sedate positive here would be this one, the alpha 3 component. Okay, so therefore, your T cytotoxic can kill your viral infected cell, tumor infected cell, if that one is being presented by your MSC class 1. Okay, the next one we have here your MSC class 2. So the MSC class 2 is being restricted among our APC or your antigen presenting cells. Example of our antigen-presenting cells, we have here the B lymphocyte, you could have your monocyte, you could also have your macrophage, and you could also have here your dendritic cells. So they are found on that. Okay, so MSC class 2 includes our HLA-D. We have here your DP, DQ, and your DR, of which our HLA-D are usually try to constitute here half of the entire concentration of your MSC class 2. And then again, that one is very important. In order for you to have a successful blood transplantation, then you need to have here compatible with your HLA-DR, especially for the HLA-DR. Okay, we have here the structure of our MSC class 2. So this made up of your alpha globin chain divided into your alpha 1 and alpha 2. And we have also your beta globin chain divided here into your beta 1 and your beta 2. Okay, and so our peptide binding clef here could be found in between of your alpha 1 and alpha 2. So that's the one that try to capture the antigen and try to present it. Okay, with our T helper, which is your CD4 positive. Okay, for the function, so this one is also involved in your antigen presentation. What's being presented by this one would be your exogenous antigen. So pag sinabi exogenous, those are a foreign antigen which enter your body. And that foreign antigen here has been phagocytized by our APC, your phagocyte. So actually your phagocyte. And after degradation, di ba, try to exocytose, ilalabas nila, and then it try to present the antigen. But when they are presenting the antigen, it should be accompanied with your MSC class 2 in order for your T helper here to recognize that one through its CD4 positive receptor. So if your T helper here could not, or your antigen is not being presented along with your MSC class 2, this one will not react on that. And therefore, it could not, okay, kill this one. And eventually, you cannot remove that one through different other mechanisms. Okay, so we have here the significance of our MSC. So number one, we have here for the organ transplantation. Okay, for the organ transplantation, so your MSC here, very important that you need to have here histocompatible between the recipient and donor to prevent here your graft rejection. And if they are not compatible with one another, it can eventually result here to the rejection of your transplanted na organ or tissues or the bone marrow, primarily through your graft versus host disease. So when we speak about the graft versus host disease, it is the recipient immune system that try to reject or try to destroy your transplanted na tissues. Hindi yung transplanted na tissue ang magde-destroy sa sa system ni recipient, but it is the immune system of the recipient that will going to reject the tissues. So second one, so they are also involved here. This one is also involved in your antigen presentation, as what I have discussed kanina. Okay, for the vaccination. Okay, for the vaccination here, so again, your vaccine that's being given to our body would have a specific target that vaccine are able to bind. Okay, so ang mga target nila ng ating vaccines will be your HLA. Okay, so pag nabigay ka ng vaccine here try to target specific HLA that would allow that vaccine here to stimulate your antibody production. So like for example, if some patient do not have that HLA na target ng ating vaccine, expected one that they will not produce an antibody. It is the reason actually why some patient, even they get vaccinated, but then again they were not able to produce an antibody. It's because they don't have an HLA. Allergic reaction, so again, genetic basis ang ating HLA because of your genetic basis ang ating allergic reaction because of your HLA. There are certain na mga HLA wherein we can inherit that one from our parents that can trigger for you to develop here your allergic reaction. Okay, so thank you.